Thank you for coming. So I would like to switch gear and talk about biological research. So first of all, why are animals used for animal research? So I'm a clinical microbiologist. I work at the Department of Microbiology. So for us, we look at infections. So they are actually important to understand a disease. For example, we have a case of pneumonia due to a new pathogen. We need to understand why it causes this disease. And this is important for us to find out how to treat it. So it's important to understand the pathogenesis of a disease. Secondly, we need to understand the bug. Sometimes we have new viruses or bacteria, for example, SARS or avian flu. And we need to understand why they cause the disease in humans. Third, we also develop drugs or vaccines that help to treat or prevent infection. And how do we evaluate the effectiveness of these treatment or vaccines? Animal research is very important. And finally, some groups actually do transmission studies. They will look at how, for example, will the avian influenza or the swine flu transmissible to human? What is the risk? And also, what is the risk of human-to-human transmission? So all these are very important biomedical questions that our animal research can contribute. Of course, whenever we think about animal research, the most important is to balance the benefits of the research and the problems with animal research. Of course, these research, they advance science and medicine. However, any animal research will cause some pain, fear, or distress in animals. So whenever we design any studies, we must think about this. When should animal experiments be performed, or should they not be performed? So I extract this from the Hong Kong U Laboratory Animal Unit. They should only be performed when they are essential. When are they essential? That the knowledge from the animal experiment will be used, the new discovery will be useful for saving or prolonging life, alleviating suffering, or combating any disease, whether of human beings, animals, or plants. Secondly, even some experiments were done before, we may think it's not true, so sometimes you have to repeat some experiments to know whether they're true or not. And all these must be weighed the scientific value against the potential effect on the welfare of the animal. So when we do animal research, we always think of 3 R's: replacement, reduction, and refinement. What is replacement? Do not do animal experiments unless it's absolutely necessary. Is there another method other than using animal experiments to get the results that we want? Second is reduction. Can we lower the number of, can we use less or fewer animals to do the animal experiments to get the same scientific validity of the experiments? This is very important. So one important discovery in the past few years is the use of organoids. Organoids are made in laboratories from single cells. So they are like organs, but they're not taken from any animals. So this is actually, they call it the model method of the year in Nature, the journal Nature Methods. In our department, Dr. Zhao Jie actually is very interested in developing organoids. So she has learned from a master, and she has developed 2 different organoids. One is the airway organoid. This organoid mimics the human airway in a 2D and also 3D structure. So a lot of for example, if you have influenza, we can test how influenza affects the airway. So the airway is part of your lung, very important. Also intestine. This is an intestinal organoid. So what do we use it for? MERS. Okay, you may have heard of the Middle East respiratory syndrome virus, which is very new in the Middle East, from camels, basically. One of the strange things is that it causes diarrhea. So we actually looked at MERS virus in these intestinal organoids. So using these organoids, we can replace a lot of animal experiments and reduce the number of animals that are required to study these pathogens. So organoids are similar to an actual organ. They are more likely to mimic the situation in humans because they are actually made from human cells. Therefore, it replaced some experiments and reduced the number of animals. The third R is refinement. We are trying to alleviate or minimize potential pain and distress and also enhance the animal well-being. So, of course, we must be careful that we always have enough food and water for the animal. At the same time, we must have something for the animal to play with. For example, this gadget here and something here. So the animals like to play with these. So these will help them to alleviate some of the pain and distress. So finally, I would like to say that good welfare equals good science. And this should be in the mind of all researchers who are doing animal experiments. Because if you are treating the animal badly, the results, not just for animal welfare, but also the results that you get will not be valid, and they will not be useful for any advancement in science or medicine. So I would like to stop here. Thank you for listening.